Okay, welcome back to Microscopy and Staining video series. This is the section on staining. And so here we will describe the stains that we will use in the lab actually. So we've kind of alluded to it in the last video. Staining is going to increase our contrast and allow us to increase our resolution. Okay, it's easy to see two different colored things than it is to see clear things. And we'll see that in what's called a negative stain or the capsule stain. Okay, um, and the reason they work is because they contain a chromophore. Okay, you probably will want to remember that, meaning chromo meaning color and phore meaning like light, basically. So light and color is how you see it. Okay, so I went over this in a video on how to make a bacterial smear, so hopefully you've seen that by the time you watch this. So when you want to prepare a specimen for staining, we want to make a bacterial smear, and then we want to denature it, denature the proteins on the bacteria so they can stick to our slide. Okay, so what do I mean by stick? Okay, so have you ever had an egg and you didn't put enough oil in the pan and your egg is all sticking to the pan and you have to scrape it off at the end? Well, an egg is mostly, the egg white is mostly protein, and you are denaturing those proteins to become white and irreversibly denatured. Okay, they are no longer how they're supposed to be, they're denatured, and they can't go back to do what they were doing. Now they're an egg white, we eat them, they taste good. But if you don't have enough oil on the pan, they stick. They're denatured and sticky to the pan. And that is what we do when we heat fix the bacterial smear. Okay, so now whatever we do to the bacteria, we rinse it off, we keep staining it multiple times and keep rinsing it off. That bacteria will actually stay on that slide. There are a couple of times where we don't heat fix, okay? When we do the capsule stain and the endospore stain, those items are too sensitive in that we don't want to destroy them through denaturing, and so we don't heat fix those, and we're just lighter on our rinses, and we make sure that we completely air dry it before on the air dryer. All right, so simple stains improve the contrast just like always. Okay, in our lab, this is what this is over here. Okay, we can use safranin, and it's going to give it a nice pink color, or we can use crystal violet, and that's going to give it a nice violet purple color. Okay, you don't want to use both, all right, because that would end up being overstained, or you're trying to do a gram stain, which we'll see in just the next slide. Okay, so what do I mean by increased contrast? Well, you can't see any bacteria in an unstained bright field microscope. You probably could try. You might be able to see some heavy stuff or some bigger bacteria, but just adding a stain for 1 minute is going to make us see everything. Okay, and we can see now under our light microscope, we can see all the bacteria. Okay, and these would be rod-shaped bacteria here, coccus bacteria here. So this is a mixed sample, and again, this is purple, but since it's a simple stain, it is not gram positive based on this image. This is just a simple stain. They're just stained with crystal violet. Okay, so be sure to read any picture inserts if you see any purple or pink items before you make an assumption that it is gram positive or gram negative. So now we actually move to the gram staining procedure. We're going to do this a lot in the lab. You're going to do it with this week and next week in the lab, and we're going to do it for our unknown case studies, and we'll keep doing it okay, throughout the semester. So the gram stain is pretty important, and you're going to want to know this for the final exam and the lab practical. So this is one of the most important things after hand washing that you're going to need to know in the lab and lecture. Okay, this one transcends both. So the gram staining procedure is we're going to flood it with our primary stain for 1 minute and then rinse it. Now, these boxes are really important. These are the conceptual boxes. Okay, you got to know them. All cells at this point are purple. They all have taken up the crystal violet, and they're all going to be purple. Their peptidoglycan is purple. Now we're going to increase that bond, and we're going to make it stronger by adding mordant for 1 minute, and then all cells stay purple, right? Because we haven't decolored anything. All cells should be purple, and those bonds should be tighter. Okay, so know that. That's what we're doing with the mordant. The mordant in our lab is iodine. Mordant in most gram staining procedures is iodine. Okay, the slide is flooded with solution of ethanol, methanol, acetone, decolorizer, you might see, then rinsed with water. Okay, this is very short. We don't want to decolorize our gram positive layers. We just want to decolorize our tiny, thin gram negative layer. Okay, um, but the gram negative cells are now colorless. So the ones that are gram negative with just a tiny layer after our decolorizer are colorless, and all the other ones are purple. All the gram positive are purple. All right, so this is the most important step, the decolorizer. Okay, and most important conceptually, okay, you're gonna need to know which cells were decolorized. The cells that were decolorized were the gram negative cells. They all were added to decolorizer, but the only ones that lost their color were the gram negative cells. Okay, so then next we're going to go ahead, and the slide is flooded with safranin for 1 minute, and then it's blotted dry. Okay, and so what this is going to do is it's now going to increase your phase contrast between the two. You will see gram positive be purple and gram negative be pink. Remember, purple positive, PP, purple positive, and then negative has an N in it, so does pink. Okay, so that's how you're going to need to know the end result. Okay, so why does this work? And we've kind of touched on it before, but this is where we really want to drive it home. The gram staining protocol differentiates between these two cells. So cell walls have many thick layers of peptidoglycan if they're gram positive. The peptidoglycan layers are commonly embedded with tectoic acid. Okay, so let's just imagine that they have layers of gram positive as big as my head. Okay, that's how many layers of peptidoglycan they have. And gram negative actually has just a tiny thin layer and then an outer membrane, an outer phospholipid bilayer on the outside. So they can actually be more selective with what they let in. Getting drugs to kill them or sanitize them is much harder because of that outer membrane. Okay, but they only have a thin peptidoglycan layer that we take advantage of. And so that is in a periplasmic space. Okay, and that outer membrane is external to the peptidoglycan. But it doesn't matter. The decolorizer gets through the membrane rather easily and decolorizes the peptidoglycan in seconds. Okay, so this is what we're talking about. As you can see, the gram negative right here has that inner membrane and then the outer membrane and the periplasmic space. Okay, and then that blue rectangle chain is the peptidoglycan cell wall. See how thin it is? We can decolorize that in 10 to 15 seconds. Okay. Meanwhile, the gram positive over there, way over there, has a lot of blue rectangles, signifying it has a lot of peptidoglycan, and that dye gets caught up in all that peptidoglycan layer, and we can't decolorize it in 10 to 15 seconds. Meanwhile, we can decolorize that pretty easily. Does that make sense? So that's what we're taking advantage of when we when we do a gram stain. And what does that mean? It means that gram-negative ones, because they have that outer membrane, they're a little tougher to combat. Okay. They also have this LPS layer, which we really want to know. Okay. Lipopolysaccharide layer. You can see that pink layer on the top. That works as an endotoxin. Okay, we'll come across this later and repeat it again and again, but you need to know this lipopolysaccharide layer. Okay, it has lipid A on it. Okay, lipid A, lipopolysaccharide layer. This is very extremely important. This is an endotoxin. This is what is a toxin for humans. If you kill this too fast, you kill too many of them too fast, all of that LPS layer is still in the person and it can be toxic to them. Okay, so it's called an endotoxin erroneously because they thought they were on the inside of the cell, but it's still an endotoxin. We'll talk about exotoxins later, but we must remember LPS layer. Okay, that's the LPS layer. Okay. And for that, we are done with the gram stain. I hope you take to heart all the gram staining information that you've been given. It's going to be highly important. If you have any questions on gram stain, please ask. We'll go over it again and again in the lab, so we shouldn't have any problems. Purple, pink, purple, pink. So positive is purple, pink is negative. So that's a purple positive, purple and pink scale. Now we're going to go to a different color scheme. Okay, this color scheme is like Christmas, red and green. Okay, so you can see in that picture the bluish green circles and then the long bacillus chains. Okay, so this is called an endospore stain, and by now you've hopefully heard of endospores. Endospores are those survival structures. Okay, and to stain them is rather elaborate and kind of being very chemical heavy. So we want to be careful with that. So in the lab, we kind of just do it a roundabout way, is that we just soak it for 30 minutes. And that takes a while, so we have to be really ready when we're doing an endospore stain in the lab. But an endospore stain is going to show our vegetative cells in pink. Vegetative means they're alive. They are growing and dividing, vegetative like a vegetable. And endospores, those survival structures, are going to stain blue. Okay, and so if all the nutrients run out and all that water runs out, those bacteria will die and those spores will remain. And then once the nutrients are returned, those spores live on. Okay, so remember red and green, or red and blue-green for endospore stain. Okay, and this will be done in the lab by you, and you'll see something very similar to that. Okay, so we'll do the gram stain, we'll do the endospore stain, and we'll do this last stain that we see here, the capsule stain. Okay, by now we've gone over glycocalyces or glycocalyx, and a really organized one is called a capsule. Okay, but we can't stain them. They're just kind of too hard to stain, and so what do? So we do what's instead a negative stain. Okay, we're going to make a negative of the image. We're going to actually stain the background by leaving some stain there on the slide, and we're going to stain the bacteria themselves. So now we kind of create a layer that we don't see through contrast, and this is the negative part of it. We are actually trying to see the capsules, but we aren't staining them. That's the negative part. Okay, so we'll be doing a capsule stain too. A capsule stain is much easier. It's like 2 minutes long, and you get to see all these cool capsules. Okay, so again, the capsule is a nice organized layer of sugars, essentially, forming that glycocalyx. That layer isn't stainable. That layer is seen, though, when you stain the bacteria and when you stain the outside. Okay, and that is what we see in the negative capsule stain. And that concludes our microscopy and staining lectures. I hope you'll join me for the next set of lectures on the prokaryotes and eukaryotes.